boring. It's going to be fun. Yeah, right now statisticians during this pandemic of the coronavirus work really, really hard collecting the data on infected people, people in hospitals, how the virus is spreading, how the virus is changing while spreading, because there are mutations happening all the time in the RNA sequence of this virus, and we can actually track coronavirus COVID-2 right now from one country to another. We can see that, for example, the strain of the virus in Italy didn't go directly from China, but it traveled through some other European country. And right now the hypothesis is it traveled from Germany, and you can figure it out from the sequence of nucleotides in the viral particle. So what we're going to do today, we're going to analyze nucleotide sequences and see if we can figure out the original virus, the ancestor for the viruses that that jumped somehow in humans. You guys have heard that the origin is probably from bats. There are recent papers that maybe it's from pangolin, that's an animal that lives in Asia. And the papers that are published on all of that, they're published like last week, literally. So we had to put this tutorial in like 3 days or something. So everything you guys are working with is super, super fresh. And also, if in 3 months there is another paper published saying all of that is not true and something else is true, updated version of all these sequences and some new origins of viruses were found, it is okay. We do not claim that we're going to solve the world's problem with this exercise. For us right now, it is just to learn how to use the available data, publicly available data, and make some sense out of it using some very, very basic software. If you're staying in biology after you graduate and you're doing some research, chances are you're going to use this software in the future, that's for sure. Okay, so today we are going to learn what is a phylogeny, and this is going to help us to figure out the origin of the coronavirus that everybody is so scared of right now, which is causing this dangerous pandemic. Okay, so phylogeny is an evolutionary hypothesis of relationships that is presented in a graphical form. So this is a graphical form and this is a graphical form. So relationships between who? Relationships between biological species or larger biological groups. Let's say a cat is a species in biology and a lion is a species, and we all know they are related because they are from the cat family. So we would say the cat is this species and the lion is this species. Long time ago, they used to be not different from each other. There was an ancestor, right? And this ancestor is now located in this node, right? And this ancestor with time has evolved into a cat in one environment and into a lion in another environment. And maybe there is a tiger and a cougar species, the CAD I just encircled, those are 2 other species. They are related and they had a common ancestor a long time ago, let's say 100,000 years ago. That was an animal, that cougar and a tiger, but with time, populations of these ancestors split and formed 2 totally different species, and now those species are not even breeding together, and you cannot get something if you, let's say, try to cross them and try to get some offspring. This is not going to happen. They are way too different from each other. All right, and this would be the ancestor for the ancestor of tigers and cougars, and for the ancestor of cats and lions. Let's say 1 million years ago, there was an animal that looked like all of these animals, and with time, this and that slightly changed, and populations of these ancestors separated and speciated into different species. All right, so look at this hypothesis too, right? This is our understanding that C is more related to D right here, and A is more related to B. But guess what? Maybe somebody has another hypothesis. Maybe somebody says, you know what, a cat and a cougar and a lion, they all are super related to each other. So they have a common ancestor in here, and then the groups have speciated, and the tiger is so far away, this guy is not even related closely to these guys. Maybe an ancestor for all cats actually existed very long time ago, but it's not as similar to all these groups as they think. So this is altogether a graphical representation of our understanding of where the species came from, what was their ancestor. And in certain types of these trees, you can actually label the time, how much time it took, how many millions of years it took for one group to form and separate from another. And this is hopefully that's something that you can remember from your general biology or even biology course that you took at school, in high school, right? So the tips, right? The tips of the branches are called terminals, so that's a terminal, and the intersections are called nodes. This is just something that we're going to use throughout the class, and those are the terms I want you to operate when we answer questions in our tutorial. Now, synonym for these three: phylogenetic tree, phylogenetic hypothesis, cladogram. We will learn what clade is in a second. Evolutionary tree. So pretty much this is the evolution of organisms that is represented in the form of a tree. It doesn't have to be a million years for the organisms to evolve. Right now, the virus that is attacking the human population is changing super fast. Strains are mutating. There was a strain in pangolin or a bat that jumped from this organism into humans just because it developed a couple of mutations in the DNA, and that doesn't take for a long time. Bacteria and viruses change super fast to compare to more complicated species, and hopefully you will see it in your phylogenetic tree when you construct one. Okay, so a bit more on relationships between groups in a tree that you can figure out. So this is our terminal, right? So the clade, right, or monophyletic group or natural group, you don't need these terms. So the clade is a group of species that includes an ancestor and all of the descendants. So this green thing is a clade. Yeah, this is a descendant, and this is a descendant, and this is an ancestor. Yeah, this is a clade. Now, I can say the clade also is this, right? Because this is an ancestor, and those are terminals, all the descendants from this ancestor. That's also okay. Yeah, I cannot say that this is a clade, right? Because the ancestor for D, right, is here, right? I need to include all ancestors of all descendants. So this is not a clade. Okay. Now, this is just a graphical confirmation of something I told before. Here, you're going to have the oldest ancestral groups, right? And with time, the groups will split into populations that will continue to speciate into smaller and smaller groups. Okay. Now, let's take a look. Here's a question for you. Is C more closely related to D or E? And I'm going to show you how to answer this question. Right, so this is my C, and this is my D, and this is my E. So I'm going to figure out. Thank you, Michael. You're so getting an A in bioinformatics. All right, so I'm going to explain to you how to answer this question, and we're going to answer a different question on a different slide. Right, so how do I know if C is more closely related to D or E? I am going to travel to the origin of all ancestors. The origin of all ancestors and descendants is going to be called a root. So this is the oldest group of organisms that belong to this tree. And A probably is going to be the very oldest group of organisms that's speciated from this root. So I'm going to repeat that. Yeah, so I'm going to travel from C to the root, to the origin of the tree. And you guys will have to root your tree when you put together phylogeny. So that tree would show you the correct relationship between the terminals. Right, and now I'm going to travel to the root from the D. Yeah, same path. And look, it took me just a little bit to find the common ancestor between C and D, and the distance is very short, and for C and D it's very short, and my ancestor is in this node, right? Very short path. Now, if I travel to the root from E, right? I'm traveling to the root from E. The first ancestor, which is common between C and D, is going to be here. This is the first ancestor. Okay, and look how much I had to travel. All right, this is a very long distance. This distance is much longer than the distance I need to travel to get to the common ancestor from C and D. That tells me that C is closer related to D than to E. All right, so C and D are close species, and C and E are much more far away from each other. So they are not as closely related, evolutionary speaking, as C and D. And this is a type of analysis I would want you to perform when you look at your own tree after you construct. I want you to explain that from your tree you can hypothesize that this particular species of a virus is related to this and this one, and it is far away from that one, although altogether they form the same clade, so they are all hopefully related somehow. So that's some kind of explanation and analysis I would want you to perform. Okay, now a couple of more words on the root. So determining the root of the tree is very important. Yeah, root lets me to select the group of organisms, or the taxon, which is pretty much the common ancestor with like super minimal amount of mutations that happen in time. And the rest of the tree then form proper relationships between the termini, and you will know which organism is close to another organism, like for real, right? If the tree doesn't have a root, like this one, this is an unrooted tree, then you don't know which organism is close to which one and which one is related to this one. And look what's going to happen if you select different groups as a root. So now I'm saying C is a root, right? C is the ancestral group. Now D, A, and B are going to be closely related to each other. But if I say D is a root, this is the ancestral group, right? Now C and B are evolutionary younger and related to each other, right? So you can convert this unrooted tree in multiple versions. And how do you know which one is correct, right? You wouldn't know which one is correct. So in order to root the tree properly, we add a sequence of the virus that is related to coronaviruses, but it's not very close enough. So it's like a control that will not cluster or form these related groups in one clade with your coronavirus, and is there for you to say this is a root. You're going to click on that sequence and say, I define that this is a root because I know for sure this is the most ancestral group that is outside of coronaviruses. So the tree should start branching from it. And then your tree will be hopefully nicely organized with proper relationships between the groups. Okay, so those are some variations of the trees. Just set an example on how the tree looks like. If this is my common ancestor right here, right, this would be evolutionary fishes, the ancestral group for everything. This is the youngest. The um, root is here, right? The root is here, and the fishes is the oldest, most ancestral group. And then look at this. We can travel through this tree. Then frogs appeared in evolution. Yeah, there was an ancestor for both frogs and altogether this clade. And from this clade, lizards and chimps and humans appeared. Now, look at this. You can rotate around the nodes right here. Okay, so you can change the position of chimps and humans, and exactly the same is going to happen, because like we already know, it's not about the distance like this between chimps and lizards and between humans and lizards, right? This tree is not telling you that chimps are closer to lizards than humans. No. We need to travel to the common ancestor between chimps, humans, and lizards. And the common ancestor is here. And you can see that the common ancestor that I labeled with the arrow is located at the same distance from chimps and humans. So whatever you put up and down on this tree as an artist, right? Chimps, humans, whatever you put in there, you are going to have exactly the same distance to the ancestor between these two groups and lizards. That means we both, with chimps, are related the same to lizards. It's not like one group is more related than the other. And at the same time, we're going to be called sister taxa because we belong to one clade and we share the same ancestor. Now, we have seen a tree like this. It's a diagonal tree. You can represent it as a vertical tree. You can represent it as a horizontal tree. All of that is exactly the same tree. It's just a different graphical representation. Your MEGA is going to show you the horizontal tree. Everything else is up to you. You can play with graphical representation and make it a circular and vertical and diagonal, but the information is exactly the same. See, chimps and humans are here in one clade, then lizards right here. It's exactly the same information. Okay. All right, so this is just, let me see the color. Okay, so this is just a repetition of what I just explained, how to find the most recent ancestor, the most recent common ancestor for C and E, right? I want the most recent ancestor for C and E. I'm traveling to the root, yeah, from C and E. Here is the node, yeah, where they have a common ancestor. Okay, so I would want you to look at the tree that you guys are going to decide to design after you work with DNA sequences, and tell me, who do you think is the common ancestor for the COVID-2, the SARS virus that is attacking the human population right now? You should be able to figure it out. If not, then speculate. All right, so I'm gonna do a quick poll. Please do not type anything in the chat. I'm gonna give you an option, ENG or ENC. All right, you're gonna look at this picture and tell me if it is ENG or C and E. Who is closely related? Half of the work is already done for you. Yeah. Zachary and Wanda. Wanda. Awesome. Okay, let's take a look. So most of you answered correctly. It is E and G. Okay, so let's go to the root from E to the root, from G to the root, right? This is the common ancestor. Very short distance to the common ancestor, right? Short distance. But if we travel to the root from C and E, right, to the most common ancestor is here in this node. The distance we have to travel to the most common ancestor is much longer. Yeah. The longer distance you have to travel, the far away and less related are the two species or groups of organisms we're talking about. So the correct answer is E and G. E and G is more closely related. And this is a type of analysis I would want you to do in your work. Okay, so there are two ways to build the phylogeny. The software is going to consider a couple of factors. You're going to do both maximum parsimony and maximum likelihood. In parsimony, the software analyzes the sequences and selects the tree that uses the fewest explanations on how this tree is generated, right? So during the evolution, the nucleotides are going to switch from C to A, from T to A, from G to C. That's going to be a mutation. So the fewest changes it's going to take to make a tree in the method of maximum parsimony is going to be the version of a tree that's going to be selected. So the tree that is built by the maximum parsimony contains the fewest number of changes of nucleotides that would result in these type of relationships graphically presented by this tree. And the likelihood is a statistical method. It's going to use a model, mathematical model that will consider the statistical chance or likeliness of, let's say, cytosine to guanine, because cytosine can change to guanine. They're both pyrimidines, right? They can change one to another easily, but it's not that likely that cytosine is going to change to adenine, right? It's less chance. So the mathematical model is going to assume that and then select a tree that is most probable according to all these likeliness of nucleotide changes from one to another. So in order to build such a tree, you will have to test one of those models. Again, you don't do any math. It's a software. You click the button and it does everything for you. Okay. Now, I don't think I have a slide. Okay. I have to type it in here because I think I don't have a slide. Yeah. Now, there is also another measure, bootstrap, that is called bootstrap. So when you are building your trees, okay, oops, okay, let's erase everything. So let me type it in here then. So I just typed really quick in web, so I don't have to retype it in here. So bootstrapping is a mathematical method that would remove like extra information from the point of view of a software from the data set that you're working with, and then rebuild the tree and look if the tree is looking the same, and then remove a little bit more data, rebuild the tree and see if the tree is looking the same, and repeat this iteration like 100 times, and this is something that you would have to ask your software to do, repeat this 100 times. So if it repeats it 100 times and the tree is still the same, then it's like 100% chance that this is a likely tree. It's a very nicely built, confident tree. And the Bootstrap support is going to give you a number, how confident is this particular design of a clade. There is a question in the tutorial that is going to be asking you how confident you are that this particular tree is making sense. So it's not just a random fantasy of your computer program. And in order to answer that question, you will have to look at the Bootstrap support. If the Bootstrap support tells you 100% chance this is true, there's a number, Bootstrap number says 100% chance I did modify this and that and still the same tree, so I'm pretty much sure this is the clade. Then you're going to say, I'm super confident this is the correct prediction of the evolutionary relationships between two organisms. But if the Bootstrap is very low, it's like 20% chance that this is the correct clade, then you're going to say, looks like it's something to discuss, but I wouldn't be sure that this is the correct prediction of the evolutionary relationships. We need more data to build a better tree and a more confident tree. Okay, so before you move on to the actual building of phylogenetic tree, you need to first download RNA sequences because the coronavirus is an RNA sequence. We have already done this for you. You will have a couple of exercises just to check what's in the public database and CBI database. Most of you already used it to search for publications, but you're going to search it for the sequences of the genes today. We have already found like 12 sequences and put them together in one file that you're going to work with. But the software doesn't really understand the similarities and differences between these sequences unless you do something that's called an alignment. Okay, alignment. Yeah. So alignment compares multiple sequences and optimizes the sizes of those sequences, the lengths, and finds the areas that are the same or homologous, right? They are the same, they are from the same origins, and the spaces that parts of a sequence that are different, and says, okay, if this is different, that must be some kind of mutation. Yeah. So I'm going to explain on the next slide what that means. So alignment in general understanding is about introducing of gaps into your sequences. So let's take a look. This is the gene number 1, and this is a gene number 2. Let's say I have two ancestral felines. They're not tiger and cat yet, domestic cat. There are some kind of ancestral felines, right? So one of them has gene number 1 and the other one has a gene number 2. Now, during the speciation, right, the two groups, two populations of these two of these ancestors separated from each other, and in one group this gene has a deletion, right? The guanine was removed from this sequence, disappeared, and the other group had this mutation, inserted GTA into this place, right? And now the populations of animals will give this mutation to their offspring. As a result, what we're going to have, right, in one group of animals, future tigers, this gene, and in another group, future cats, this gene. Yeah. The software would have no idea what's the difference, what's the similarities. It would treat it as like a look at the colors. CC, AT are exactly the same, and they are from the original genes. And CA, right, AG, TCC, the orange, they are also the same. Yeah. They are from this gene. So we need to tell the software those are actually similar pieces of the genes. You have to treat them as if they're similar, not totally different. So the software is going to perform this alignment. It's going to find the pieces that are labeled with the same color on this illustration. Of course, there will be no color when you feed the sequences to the software, but in MEGA, when you perform the alignment, you will see that actually it colors it for you. Yeah, and instead of this insertion of GTA, the software is going to insert those gaps, right, to fill in the empty space. And it's going to insert another gap to fill in the missed guanine that it lost earlier, right? It lost it in here. So now this is aligned, and now the phylogenetic algorithm that builds the tree can look at it and say, oh, okay, those are actually very, very similar trees, and what happened is just three very similar sequences. What happened is just three nucleotides disappeared from one sequence and one nucleotide disappeared from another, and that's it. Now the tree that you're going to build is going to be much more accurate and much more reliable. So that's an alignment, and again, you're going to download the file that is not aligned, just sequences all together. You're going to press a button and get yourself an aligned file, and that's the file that you will have to use to build the phylogenetic tree. You will save it in a weird format for MEGA. You will name it coronavirus aligned, and that's the file you will have to use for the tree, otherwise your tree. All right, so we're almost done with the introduction. So what can we learn about the SARS-CoV-2? This is the official name for the new virus that causes the COVID-19 disease. Viruses reproduce really. Emily has a question. Do we introduce a gap if there is a point mutation? Your software will do it for you. You feed the file with multiple sequences, and the software will introduce gaps where it thinks the gaps should be introduced. You don't do it yourself. Okay, it's all software. So viruses evolve super fast, right? They reproduce super fast, much faster than animals or plants. So you can see the evolution within like a couple of years. There is a new strain already, or even a year right now. Boom. But the genomes are small, and in professional research setting, those are actually complete genomes that are going to be compared. What we did, we trimmed those genomes for you. You're going to use only one gene, Rna-Dependent Rna Polymerase. I'm going to type it right here. Rna-Dependent Rna Polymerase. Yeah, that's the enzyme that's encoded in the RNA of the virus that can read the RNA and synthesize a complementary RNA strand to produce more RNA viral molecules, to assemble more viral particles. This is something humans do not have. And this is something that we produce for virus. The virus infects us with its RNA and occupies our ribosomes. Ribosomes are going to use the viral RNA to synthesize a protein, and that's going to be the enzyme RNA-dependent RNA polymerase that will start making multiple copies of the RNA for the virus, and we're going to also make our with our ribosomes and everything the protein for the virus. The virus is going to self-assemble and infect somebody else. So they are pretty much parasites using our cellular structures to copy their own RNA. So this is a shorter version, and your programs are going to work much faster because of that, because if we give you 12 full-length genomes, you're not going to ever finish with this. Now, once we figure out the origin of this gene, we can and the whole genome, right? We can see what kind of mutations happen during the evolution of the virus. We can predict, if we know the sequences from of similar viruses from other animals, we can actually predict from which animal it came from. We can figure it out where it came from. We can see what kind of antibodies other organisms develop to fight with this. So there's a lot of health-related science that can be done once we perform very basic biochemistry and bioinformatics, and that's something I want you guys to play with today. All right, so you're gonna get a file with sequences of the RNA-dependent RNA polymerase for similar coronaviruses. SARS-CoV caused the SARS epidemic in China in 2002. MRSA caused the Middle East respiratory syndrome in 2012. And you're going to also get sequences from other animals such as bats and pangolin and see if they are related to what we are working with. And there will be one sequence from unrelated virus that you're going to use to root your tree. It's there just for the purpose of rooting of the tree. All right, so the software you're going to download is called MEGA. It is free, and we already quickly discussed how to download it, but your tutorial also gives you some recommendations, and I'm going to repeat again. Please download an older version if you're using Mac. It looks like the older version works with Mac much better than the Megas. And the sequences you guys receiving in a file are from NCBI. And first couple of questions in your tutorial will ask you to check those sequences so you actually know where we take them from. We're not inventing them. We're not doing the sequence by ourselves. All these sequences are super fresh. Some of them were uploaded to NCBI literally like a couple of weeks ago, so they're not annotated very well. And every day more and more sequences are uploaded. So again, like I said, in 3 weeks, we might have a totally different phylogenetic tree because we know more of sequences of viruses from, let's say, some other animal populations. But it is what it is right now. This is the module we designed for T. So today we're going to play with the S protein from coronavirus. It's a spike protein. It's a protein that is located on the surface of coronavirus, and that's the one that is going to contact ACE2 receptor, acetylcholinesterase 2 receptor on our cells. It is definitely not designed for the virus. It's designed to interact with some hormones, but the virus figured out how to attach to it and then enter the cell. So our job for today is to learn how to model this protein, how to figure out its 3D structure, and label important parts of it. So that's the job that's done by biochemists and computational biochemists. Virologists, they would think of epidemiology of this and the role of this protein in the viral entry into the cell. Fundamental biochemists, we have to figure out the structure of a protein and see how it can be used in prediction of the activity of the virus or creation of antibodies or something like that, or vaccinations. Okay, we remember this, right, very well. So the protein we're going to work with today is going to be one domain of a more complicated spike protein. I did design this exercise in such a way so you don't have to download quaternary structure because it's really hard to switch between all of these subunits trying to find which one to work with. So we're going to download the amino acid sequence only for the one subunit of a pretty complicated protein of coronavirus. So who cares about the 3D structure? Right, we can predict the 3D structure using computational software. We know about it and we have heard about this in our classes, but it's never 100% guarantee that the prediction is correct. And if we want to understand the biological function of a protein, we need to know how exactly the tertiary structure is designed. Once the exact folding of the protein is known, we can see which part of the protein is sticking out, which part is interacting with the receptor. Okay, if this part is very important and it's kind of available for the antibody to target it, then that's probably we need to select for the antibodies that will be able to interact with this part of a protein, for example. Okay, we already had presentations a couple of weeks ago. There are two main methods, NMR and X-ray crystallography are very good presentations that have discussed these two methods that will look at the protein that is first crystallized for crystallography, right, or prepared in some other way for NMR. The methods would require very pure protein. The purity of the protein can be achieved via chromatography, and we will have to separate the native protein extraction like we did with beef. We extracted all the proteins from there, right? And we purified it with chromatography. Just we would have to use much more sensitive chromatographic method. We can purify it, yeah. Or we can actually insert an artificial gene that encodes this protein into, let's say, E. coli or a fungus, yeast, and ask them to synthesize a lot of this protein. It's not like we're asking them, they will synthesize it if we put it under the proper promoter. And then it will be much easier to purify such a protein from bacteria or yeast, or sometimes even plants are used for that in certain cases. And then again, this pure protein can be used for identification of the 3D structure. Yeah. So a couple of words on X-ray crystallography. That's the major method that's used to figure out the 3D structure of the protein, and that's the method that was used to figure out the structure of S protein, the one that we're going to work with today. You hopefully remember, or maybe yes, maybe not. If you prepare that presentation, then you definitely know. First, we need a crystal of this protein. It shouldn't be a soluble, soft molecule. It has to be a crystal. And figuring out the conditions for crystallization is a lot of work. People spend years trying to figure out what's the pH, what's the temperature, what are the other components of the solution for crystallization that have to be present. And they change pH like 1 decimal point at a time, but everything else is the same, and see if the protein forms a crystal or not. They sometimes use, scientists say, they sometimes use other chemicals, polyethylene glycol, for example. It's a viscous polymer that would facilitate crystallization of the protein. So once they get a crystal, it is just a matter of time. Something that called a synchrotron is used. It's a machine that delivers the X-ray to the molecules, to the crystals, and then, the CCD cameras, usually the digital cameras, are going to read the absorbance versus transmittance of these X-rays and record non-absorbance as little dots. And the crystal is going to be rotated, and then something that's called a goniometer is going to be used, and that goniometer is going to measure the angles between atoms. Then the software, lots of math and lots of bioinformatics, is going to create the density map for electrons, and then another software is going to build the model that this part corresponds to alpha helix and this part corresponds to another alpha helix, etc. And the coordinates for the atoms in such a model are going to be deposited in the protein database or PDB database. It is a publicly available database, same as NCBI. And crystal structures of all proteins that are publicly available can be found in there. Okay. Now, if you guys have time, hopefully yes, I suggest you watch this movie. It's called Naturally Obsessed. So it is a movie about several graduate students who work in a protein crystallization laboratory. They are crystallizing some important proteins and are trying to get their PhD, and it's probably not an hour. But you can see how the grad school looks like and what are the challenges in grad school, and not all of them are going to graduate. So if you are interested in continuing your career in biology and getting a more advanced degree, I highly recommend you to watch this. Or if you're just wondering what's happening in graduate schools, how people work on PhD, what the students do, and how the crystallization happens, that gives you some kind of idea as well. Okay, so the Worldwide Protein Data Bank, or PDB, is the major protein structure database, and you guys are going to retrieve the file with amino acid sequence from this database and with the atomic coordinates, so your software PyMol will be able to arrange it in a 3D model. So everything about the protein, how the protein was expressed, what are the crystallization conditions, and all of that is also deposited on PDB. And you can go through, figure it out. It's just right now, so many coronavirus proteins are deposited. Sometimes the proper annotation is missing, but with time, the authors hopefully will update it with proper annotations. So there are other databases for NMR, for other chemicals. We don't really care about them today. We will work with the protein data bank. Okay, now there are also many programs that will allow you to explore the 3D structures of your molecules, and the PyMol right now is one of the most widely used. It is open source, it is free for educational purposes. In 25% of all publications with protein structure, people have used PyMol. So Py stands for Python. Since it's free, it's not extremely user-friendly. You will need our manual, and maybe you will have to watch an instructional video on a different protein, on how PyMol looks like and what are the commons that can be used in this PyMol. But your tutorial will take you through the steps same way it did with MEGA. Yeah, you should be able to complete it just with a tutorial without the video, but please go ahead, watch the video if you're not very good with computers and software. Right? But this is a very, very classical software, and it's used in pretty much any lab that works with proteins. Okay. So your protein at the end in your image is going to have parts that look like a cartoon. It's going to have a small part that I will ask you to label as stick. Yeah. And there will be an SE2 receptor part that I will ask you to represent as a solvent accessible surface model. All of that is written in your tutorial, how to select a part of a protein, how to present it as a cartoon, how to present it as a stick model, how to present it as a solvent accessible surface model. And I'm here till 3:15. Please do not forget about this, because that would be sad, right? I gave you a lecture, you left, and then in a week you started using the software and you have a question and it's already too late. All right, so this is something that I actually discussed at the beginning of the lecture. We are going to use a model of S protein. This is from the current SARS-CoV-2 virus. And this is a protein that the virus is using to enter the cell. It will interact with ACE2 receptor, and then a couple of proteases are going to cleave the virus, and the virus, sorry, not the virus, the protein, and the protein is going to be cleaved, and the virus will get into the cell. So if we understand how this protein is designed, we can find the critical points, critical amino acids that are required to interact with this receptor. We're going to hopefully find the parts that work as antigenes for our own antibodies, so the antibodies can recognize it and eliminate it from the body. We can probably design a vaccine that would expose these parts to our immune system, so the immune system will start developing antibodies before the virus has a chance to enter our organisms, right? So when it enters the organism, we already have antibodies to protect us from it. It's not going to invade the cell. We don't have to wait for a week or two to create those antibodies. All right, there'll be a couple of questions. It's not only step-by-step instruction. There are no questions like in MEGA that you have to type, but there are things like label this and label that. And from your image that you generate, I will understand if you understood the question.